All right, so good morning, everyone. Thanks for joining. I'm Markita Landry. I'm an assistant professor in the Department of Chemical and Biomolecular Engineering at UC Berkeley. And today I thought I'd take a bit of a zoomed out view. I have, I think, one slide of our lab's data, but to bring into the conversation the use of abiotic materials, specifically nanomaterials, for genetic manipulation in plants, though certainly these materials have also been used for the delivery of nutrients and pesticides at a rapidly growing pace in agricultural biology. So the motivating factor for a lot of the work that we do, especially for genetic modification of plants, is in the projected dearth of nutrient-rich food, especially in the light of population growth and a decrease in the availability of arable landmass. So specifically, projections estimate that food production must increase by 70% in the coming decades so that we can feed our growing global population. Now, genetic engineering of plants is one of many possible ways of addressing the potential shortcomings that we're about to face in our food supply. And so within the context of what's currently being done in genetic engineering of plants, the tools that exist for introducing foreign DNA, RNA, and proteins, which are necessary for the genetic manipulation, are twofold. And they're depicted here, the first of which is a biotic method, so specifically agrobacterium-mediated delivery, in which we take advantage of a pathogen-plant interaction in which agrobacterium can be genetically manipulated at the bench to contain a gene of interest that can then be transfected into plant cells, integrated into the plant host genome, and producing transgenic plant tissue that can then be regenerated into transgenic plants. Agrobacterium-mediated delivery suffers from drawbacks of species range limitations. So some species, especially wild species and even subcultivars of the same species, might not be susceptible to agro-mediated infection. Conversely, biolistic particle delivery is an abiotic method whereby DNA, RNA, or protein can be dehydrated onto the surface of a gold particle and can be shot into plant tissue using a high-pressure gene gun. So the advantage here is that we have a method that allows for cargo-independent delivery, whereas agro is largely confined to DNA delivery alone. The gene gun method, however, usually suffers from low transformation efficiency and cell damage due to the biolistic nature of the delivery, which can pose some challenges upon regeneration. So with both of these methods, there's a lack of control, both over the frequency of transgene integration, especially for biolistic delivery, and also the location, which could potentially disrupt endogenous genes through integration of the transgene or other parts of the plasmid in the plant host genome. So these drawbacks aside, genetic modification of plants has generated still a lot of success. The United States is one of the global leaders in the production of GM crops, which are listed here. And a nice international example that I like to give is the development of golden rice, in which the gene pathway for the production of vitamin A was synthetically, genetically reconstituted into rice, so that this essential vitamin could be endogenous to a transgenic strain and then disseminated to communities for which this vitamin was found to be deficient. Now, again, despite the advances that we've made in generated GM crops, today the average GM crop still requires about 13 years of R&D at a cost of nearly $140 million, 40 of which are regulatory. So there's a significant time and also financial burden with going through a GM regulatory pipeline, where at least in the United States, GM crops are classically defined as those that have been generated through biotic or pathogen-based methods for which there is integrated foreign DNA into the host genome. So part of what my lab works on is in developing nanoscale tools that are able to efficiently deliver DNA, RNA, and hopefully in the future proteins as well in an efficient manner, in an abiotic manner that would be plant species independent and ideally tissue independent as well. And lastly, one that has greater control over the integrating nature of the plasmid. So non-pathogenic and non-integrating with an eye on the regulatory side of things.
1 minute left.
And so here, I'm showing some recent results for both the delivery of DNA, which allows us to express a foreign transgene across multiple plant species, and conversely, a separate platform in which we can deliver small interfering RNA into plant tissues to knock down, in this case, a transgene, but also endogenous genes. And part of the features that we've observed for both platforms is in the transience of the expression or of the gene silencing in a way that we can also confirm a lack of transgene integration, such that when we combine these tools with CRISPR-based delivery, for example, of plasmids or of the RNP itself in the future, we'd be able to genetically edit a plant, perhaps avoiding transgene segregation that is currently necessary with other tools. Lastly, to get a sense of how the regulatory map is moving within this space, GMOs have been differentially regulated across the globe for decades, ever since they became a part of our food supply in the 1980s, and how non-transgenic genome edited plants using CRISPR technologies and others will be regulated, are also starting to show heterogeneous patterns of adoption depending on local regulatory bodies. So in combination with the use and increased use of nanotechnology and plants in agriculture, I think these are also important conversations to have. So lastly, just a brief acknowledgement of the lab. Guzda Demer did most of the work that I've shown today. Also, Huan Zhang, who's a postdoc in the lab, and I look forward to our discussions in the breakout sessions.